We are very pleased to have with us Ulf Hannelius, President and CEO of Diamyd Medical. The title of his talk is Decoding the Future, Leveraging Biomarkers and Antigen-Specific Immunotherapy for Type 1 Diabetes. A little bit about him. He is a PhD, and he is going to be discussing the complexity of T1D when it comes to age, sex, genetics, and incidence, and how this needs to be taken into account when designing clinical trials and analyzing outcomes. He's going to be highlighting the Diamyd experience and data to show how this has impacted Diamyd and how the company is moving forward. Thanks so much, Ulf, for joining us, and especially in, just prior to the ADA. I'm sure you have a lot going on, so appreciate you taking the time to share this with our scientific audience.
Well, thank you for that introduction, and I'm very glad and happy to be here and to present. I'm actually bringing my family to ADA. Well, we will have a few days of holidays before ADA, so hopefully they are doing all the packing, so I can be here presenting.
Very
We'll
nice.
see. And I will, I will today present, which hopefully will be interesting. It will obviously be centered around our main asset that is currently in phase 3 trials in type 1 diabetes, the GAD-based immunotherapeutic that we call Diamyd. But the presentation will be based on some data from our previous clinical trials that I think partly I want to present how we believe now, when we unraveled all that data, we can explain why our previous phase 3 program failed and why we hope and believe that the current phase 3 program will be successful. It's all about the intricacies of type 1 diabetes as a complex disease and how you need to understand those, how the different factors interplay and how you need to target your therapeutic in order to really see the efficacy. And I assume we will take questions at the end. It will be easiest to do it that way rather than do the presentation. Yeah. So let's go to the first slide here. And here I want to it's very high level. But again, just to illustrate that type 1 diabetes is a complex disease, and there are many factors that affect the incidence of the disease and the risk of the disease. And we know that sex, gender is definitely one of those factors. That there's a slightly higher incidence in males, although it's quite equal between the two sexes in type 1 diabetes, whereas other autoimmune diseases have usually a bit higher difference between the two sexes. But it's still, in type 1 diabetes, there is definitely a slight male dominance in the incidence. Age is also a factor. Highest incidence is in the young teens and adolescents. That's where you have the highest incidence. You can, as you know, you can get the disease almost as a newborn, and you can get it when you are 99 years old of age. But again, the highest incidence, the peak, is that during early puberty. HLA, we know, is the most common genetic risk factor for the disease. So it's, as any complex disease, genetics is one of the main components besides environmental factors. But out of the genetic components, HLA, that area of the genome, is definitely, as for any autoimmune disease, the most significant genetic risk factor. And then geography. We know that there are significant differences in the incidence of the disease depending on geography. For example, Finland, from where I'm originally, that's the country with the highest incidence of type 1 diabetes, and the Nordic countries in general have very high incidences. And then there's usually like a north to south gradient. The more south you come, the lower the incidence. But then you have outlier populations. For example, Sardinia has a very high incidence of the disease, and there are certain countries, for example, in the Arab countries like Saudi Arabia, Kuwait, have high incidences, whereas some of the other neighboring countries have very low incidences. Some of that could be underreporting, for example, but there's definitely a geographic component here. Many countries, like, for example, in Japan, I know someone jokingly said that there are more type 1 diabetes researchers than patients in Japan, but you have a different form of type 1 diabetes, which it's what we call LADA, latent autoimmune diabetes in adults, and it's also called, I think, slowly progressing insulin-dependent diabetes mellitus in Japan, which is again much more common. But again, there's a difference in the type of disease that you see in these different countries. And then lastly, we also know that autoantibodies, the type of autoantibodies and the number of autoantibodies is also sort of determines the risk and the rate of the progression of the disease. But to complicate things further, we know that there are complex dynamics between all of these factors, and there are probably many more factors than what I have mentioned, but it's not like these are independent, isolated risk factors. They sort of depend on each other. And that is something that I want to, in this presentation, with examples from our trials, and show that you need to understand that some of these can be confounders in your analysis, that you need to adjust for them, whereas others might be mediators so that they work in the disease path. They are sort of might mediate the effect, for example, of your therapeutic, whereas others might just confuse the results of your analysis if you don't know how to adjust for those or stratify for those factors. And for you who have followed our company and our journey, you know that we have been in the phase 3 trials previously, and here is the New England Journal of Medicine publication from 2012, which was then summarized the previous European phase 3 trial that did not meet the primary endpoint, which was then C-peptide, stimulated C-peptide. And you can see here on the right, you see the 15-month results for the 2 active arms. It was 2 or 4 injections of our antigen-specific immunotherapeutic, which is based on GAD, and then the placebo arm. There was no statistical significant difference between the arms in preserving C-peptide. And the question was obviously why, because previous trials had been looked positive, no problems with safety. Now we know that seems like HLA is completely central to the therapeutic response of Diamyd. So if you have the right HLA type, you seem to have a high likelihood to respond positively. And the picture to the right, it's a very simplistic mechanistic picture, but when you treat therapeutically with an antigen, it's taken up by these antigen-presenting cells. Pieces of those antigens are then bound to the MHC receptors that are coded by the HLA part of the genome, and then the T cell receptors sort of recognize that combination of the receptor and the antigen bound to it. So it makes sense in retrospect if you treat different parts of the antigen are bound to different MHC receptors with different affinity. So if you have the right kind of receptor, then you will get that kind of immune response that sort of translates to a clinical positive response. This was then published. We did a big meta-analysis in 2019 that was then published in 2020, where we retrospectively tested this in our clinical data, where we had the hypothesis that HLA influences the response, and that analysis showed that there was a significant influence of HLA on the therapeutic response, and all the clinical response positive seems to be in the individuals that carry a specific HLA type that I will come back to. And here is another updated meta-analysis that was published in 2022, where we also wanted to see the correlation between C-peptide to preserve endogenous insulin production with A1C, so glycemic control. And in the picture on the left, you can see on the x-axis, you have the more further to the right, the better insulin production or preservation you have compared to placebo. And the lower down you are, the better your A1Cs are. So you want to be in the lower right corner. And you can see that here we have then looked at 3 different clinical trials in Europe and US. And you can see that the individuals that have the HLA-DR3-DQ2 haplotype, they are all in the right lower corner, whereas the individuals who have been part of these trials who don't carry that one, they are in the right or wrong side of the graph. So this quite clearly also shows both that you get the C-peptide response, but also that the C-peptide response correlates very nicely with the A1C, which in itself is a very important result, which we now know also there's a big Lancet paper that was published late last year with much more data than this that really clearly shows how preserving endogenous insulin production leads to better glycemic control on top of standard of care. So it really shows the value of preserving C-peptide. But then if we go back to the previous phase 3, there were a lot of like post hoc analysis that this is taken from the New England Journal of Medicine paper, testing like the different baseline criteria, stratifying and trying to see like how the response was in these different kinds of subgroups. It's very small here, I will soon zoom in, but you can see in general everything to the right of this dotted line, that means that the active treatment worked better than placebo. And if it's to the left of the line in this tree plot, then it means that placebo worked better or there was a negative response. But almost all of these graphs you can see are trending to the right, which shows that it was definitely not completely zero here, negative. But there were a lot of hypotheses around why the phase 3 failed. So if we first zoom in on this part, you can see that it seems like there was a significant positive response in male patients, which at that time, the speculation was that it does make sense, especially in puberty, the immune systems of boys and girls, they develop differently. And there's also this difference in incidence between the two sexes in autoimmune diseases, so that it could definitely make sense that you could have a different response to a therapeutic, but it's still a very kind of a difficult hypothesis to move forward with as a company, for example, saying that from here on we will only treat boys. It was a very difficult thing to actually motivate how to move forward here. But what we now know, if you look at data from 4 randomized clinical trials, this is in total 656 individuals, and out of these 50% roughly have the correct HLA as we know today. And here you can see then we have divided into males and females and the different age groups. And then on the y-axis you have the absolute number of individuals. You can see here in pediatrics that you have a slight predominance of female patients that carry the HLA-DR3-DQ2 haplotype. When you go into adolescence, which is from 12 years up to 18, it switches. Then you have like the males sort of have more of the HLA-DR3-DQ2. And the same trend you can see in adults, even if you have fewer patients in absolute numbers there in our trials, you see the same trend. So that means that there's actually in the age population that we had in the phase 3 program then, which was mostly then these adolescents, like there was an enrichment of the DR3-DQ2 haplotype in male patients. And this is most likely the reason why you could see in that subgroup analysis that it worked better in males. So it's not because of that you are male, it's because you happen to have more individuals in the male population that carry the right HLA. So that's one learning point, that how genetics matter really. It's really complex how the frequency of this HLA type sort of changes with age and sex. Then if we take another look here, there was also this geographic difference. So if you were a patient from a non-Nordic country, there seemed to be at least a nominally significant positive efficacy of the treatment, whereas there was no effect in patients from the Nordic countries, which in this case was Sweden and Finland. And the thinking then, there were like speculations about, well, could it be something with, for example, vitamin D and the seasonality, because we know that type 1 diabetes is also there's a seasonality when the disease is diagnosed. and also D vitamin seems to be quite important in this disease and many other diseases. And sunlight and all of these things. Here in the Nordics, we have a huge amount of sunlight in the summer. That one day we have summer, and then it's darkness the rest of the year. So maybe that could have been something behind this finding. But now if we look again at the same data set that I used previously, I look at the incidence of the HLA type that is now associated with positive response, we can see that there's a quite significant difference in the frequency of this HLA type on a geographic scale, where the southern European countries here have a much higher frequency of the HLA type, whereas the Nordic countries here, Finland and Sweden, have a lower type. So again, this is most likely the reason why we saw a nominally significant effect in non-Nordic countries, only because those patients were more likely to carry the HLA type that associates with positive response. And these are data from the ongoing trial. This is screening data from a total of 239 individuals that have been screened, I believe, in January this year. So we have many more screened today. But here you can see again, especially Spain, which is the big, what would I say, light brown, peach colored bubble. Again, a 65% frequency, 65% of the screened patients carry the right HLA type. Again, that's very much aligned with the previous phase 3 data. And Sweden is, again, lower here, around 47%. So it really sort of confirms that geographic difference in the HLA frequency. And this makes sense because, again, HLA, as you may know, it's also used as a population genetic marker because it's very variable across the globe. But at least in Western country, Europe and US, very consistently we can see across the board that at least around 40 to 50% of patients, of newly diagnosed type 1 diabetes patients, carry this specific HLA type. But then within Europe, and most likely within US as well, different demographics, there's a variation. You need to know about that when you design your trials, especially if you target a specific population. And the reason then why does this specific HLA type sort of seem to correlate with response to our therapeutic? And you may have read this paper about the endotype concept in type 1 diabetes, which is basically this, that could be that type 1 diabetes is not like one single disease. It's definitely a complex disease, but maybe you have some kind of like endotypes of the disease that lead to the same under the umbrella of type 1. So the symptoms are the same, but the pathway to the disease could be different. And there is potentially one which is called this insulin autoantibody first disease, where a person who starts their journey towards clinically diagnosed type 1, they first have the first appearing autoantibodies against insulin, and they tend to have more, they are more likely DR4DQ8 positive, so they have that specific HLA type. And it might be that there is an enterovirus that could be one reason behind triggering this disease. And it's more in the very young ages, you have this high peak of incidence of these that have the insulin autoantibody first, and then you sort of have the prevalence of that population sort of goes down with age. Whereas if you have GAD autoantibodies as the first appearing autoantibody, comes a bit in later ages, but then it's more persistent in older ages. And these are individuals that are more likely to carry the DR3-DQ2 haplotype, which is the haplotype we are targeting. And then obviously it makes sense in retrospect. If you treat therapeutically with a GAD-based therapeutic, you should target the individuals that seem to have primary autoimmunity against GAD. So that's a very simplistic rationale behind why we believe we are now targeting the correct subpopulation of patients. And this is also a very quite interesting picture. It's again, it shows from this is from our phase 2b trial, where we also recruited both HLA types. So we started that trial before we knew about this finding about the HLA, how HLA influences the effect. But here you can then see that if you are DQ2 positive, so again, these are heat plots from 3 months from baseline, 6 months, and 15 months from baseline. At 3 months, you have received your 3 injections of GAD. And there you can see if you are DQ2 positive, so the responder, clinical responder group, you see like a very immediate effect that these cytokines are upregulated, which is highlighted here in red, whereas the DQ2 negative population, you don't see any kind of cytokine reaction. And this is then GAD-specific cytokine secretion. In placebo, you see nothing as expected. And then at 6 months, there you can see that the DQ2 negative patients sort of start to get that cytokine secretion, but it's like much later than the responder group. And then at 15 months, they're sort of equal. But this is one way of also showing immunologically that the responder group, they seem to respond much faster to the GAD injection than the non-responder group. So there's a different dynamic between the two populations. And then this is then the phase 3 trial that is ongoing currently in Europe and US, where we pre-screen for HLA, which is the big difference from the previous program. There are 2 big differences, but this is the major thing, that we are really pre-screening for HLA. That's the first thing you do if you are a patient diagnosed within 6 months between the ages of 12 and 29. First thing you do is to pre-screen to check if you have the right HLA. Around 40-50% will have the right HLA, and then you move into the others, check the other inclusion criteria, which you usually pass because then it's like the standard stuff, having enough C-peptide, or GAD autoantibody positive, but with which most patients with this HLA are, and other basic things. And then you get the 3 injections of GAD 1 month apart. You also get vitamin D during this period, just to make sure that no one is vitamin D deficient, which could potentially affect the immune response, like tolerogenic immune response to an antigen. And then you are in total followed for 2 years in this trial. Again, this is what I want to highlight again here. What we are really doing is that we are only targeting a specific subgroup of patients that carry an HLA type, and that's what we nowadays call precision medicine, which has been the success recipe in the immuno-oncology space, which is becoming routine there. But I think that it will become much more routine in any complex disease, and including autoimmune diseases like type 1 diabetes. And here is the specific area in the HLA area genome, part of the genome that we are targeting. So you need to be positive for the DR3 HLA and DQ2 HLA, which is a haplotype that is inherited as a block then. But the HLA genome, part of the genome in itself is quite a large area, which is central to how the immune system sort of reacts to foreign and endogenous particles. And these are also then data from the ongoing phase 3 trial, screening data from January this year. And here I just want to highlight that it's still very consistently about 50% of the screened individuals carry the right HLA type that you can see here on the right. And out of those 50% that are positive to DR3, DQ2, about 50% of those carry the other common haplotype, the DR4, DQ8, and 50% of them are absent. And this is interesting because we also have seen that it might be that we have a super responder group within our responder group, and the super responder group are the patients who are positive for DR3, DQ2 and negative for DR4, DQ8, which could make sense because if you are DR4, DQ8 positive, you are also potentially likely to have primary autoimmunity against insulin as an antigen, And then if you sort of remove those heterozygous individuals, you have a more pure GAD responsive patient group that seems to respond even better than the heterozygotes. And also in the phase 3, we are sort of thinking about the future, and that's why my title to this presentation was called Decoding the Future, which also sounds quite cool, I think. But it's the patients who are part of this trial, we also sequence the HLA part of the genome. So we use next generation sequencing, so we really get the high resolution data. And the thinking here is that we hopefully, with a positive phase 3, would be able to also sort of start digging even deeper underneath the surface to see if we can find really super responder patients based on additional genetic factors, or if there are like individuals that could maybe benefit from a booster dose, which we have already evaluated in a few patients. So this, I think these are very interesting and valuable data to have, obviously, if you have a positive phase 3 trial, so that we don't have to at that point start thinking about how can we personalize this treatment further. We will already then sit on that data. And then just my last slide is just to highlight some of the progress that we are quite proud of and that we have done recently. Obviously, a year ago, a partnership with what is now called Breakthrough T1D, formerly JRF. So we have a partnership for the phase 3 trial, which we are very happy about, where JRF both supports us financially, milestone-based, but also obviously with all of the awareness we can get and the help from JRF, Breakthrough T1D. So this is a big thing for us as a company, and very happy to work with Breakthrough T1D here. And in September last year, we could also announce that the phase 3 started in the US, so it had been ongoing in Europe already for a while, and started in September last year in the US. And in February this year, we got fast track designation from the FDA, which is a significant boost for us, and which obviously opens up even more kinds of interactions with the regulatory agency and potentially also around these endpoint discussions, as you know, around C-peptide as the primary endpoint and potential accelerated approval pathways. But this is obviously it's a big quality stamp, and this also is good for the whole field, but it really shows that the FDA has recognized type 1 diabetes as an indication where there's a significant unmet medical need that needs new therapeutics. So it's an important designation for the field as well. And it's a very interesting summer also ahead of us. In beginning of August sometime, we will have the results from an interim analysis. It's a blinded interim analysis, but it's an important milestone because that will sort of give an indication if the phase 3 is still on the right track to have a possibility to go all the way to show a positive result, or if the other outcome will be that it looks like, based on the data that has been collected so far, that it's not on the right track, obviously. We are hoping and we believe that it will be positive, but you can never take anything for granted in this space. We know that from before, but so it will be definitely an exciting summer ahead for us that we are looking forward in the middle of our vacations. We will get that information. And with that, I will also hand over to Elna here. He will say a few words, but also want to thank, there are a lot of people that have been involved in all these data and insights. Obviously, Johnny Ludvigsson, who is the coordinating investigator for phase 3 and has been with us for almost all our trials we have done, and many other researchers, and obviously all the investigators that have taken part in our clinical trials, and the patients, and then our scientific advisory board that's headed up by Mark Atkinson. But with that, I'll hand over to you, Elna, for some encouraging words about our phase 3, I assume.
Thank you,
You're
Ulf.
on mute.
Okay, that does make a difference. I don't have any slides or anything for anyone. I just want to put out kind of what I call call to action. So any of you connected to type 1s or intersecting with anybody who intersects with, newly diagnosed type 1s. We just need to get the word out there because, as you can see from all that Ulf has said, we have to screen twice as many to find those 40–50% who have the specific HLA genotype. So I'm just putting the word out and asking if you happen to hear of them, or in any circles where they are as well, or academic institutions where you work. We have on our website, www.diagnode3.com. If you just Google Diagnode3, you'll always find it. It'll show you our sites in the US and then, of course, in 8 other European countries. So just trying to get the word out, and anything anybody can do will be helpful. And we will be at ADA, booth number, you'll find us. I think it's 823. So come find us in the exhibit hall, please. We would love to see you there.
Thank you, Elna. And yeah, hopefully the clinicians that hear this will disseminate the information to their colleagues.
It would be helpful.
Yeah, great presentation. I guess I would also say, it's sort of along the same paths of T-Shield. They also came across these findings that their best outcomes were HLA specific, right? So, and now this is the second drug that's showing this. So it's really getting some strong evidence to support the fact that these endotypes that Manuel Battaglia et al. Introduced in Diabetes Care 2020 really are showing to be something very important in terms of, if you want to talk about endotypes defined by genetics. So that's it's pretty exciting. I think it's great that you didn't just stop at a roadblock, but really sort of dissected the data and learned more and then moved forward. So kudos to you on that. Here's a question from the audience. Thanks a lot, Ulf. This is from Manuela Battaglia herself. Thanks a lot, Ulf, for this great overview. Very informative. Diamyd is a perfect example of a wise medicine developer who designs new trials based on in-depth mechanistic studies on previous studies. I often see mechanistic studies that are then not used to design more precise studies. Do you think we can start designing adaptive trials that, with in-depth mechanistic analyses, can inform during a trial how to enrich for super responders rather than wait to the end of the trial? Maybe impossible from a regulatory perspective. Not sure. What a great idea. What do you think?
Well, I definitely think it's possible. I mean, adaptive trials it's probably obviously in the phase 2 setting. In phase 3, or maybe you can do like a phase 2, phase 3 trial, but definitely in a phase 2 trial, I think it should be used much more now when we know much more about type 1 diabetes, that you can exactly using an adaptive design, if you have a good, especially if you have a good early biomarker. But we also know now, especially with C-peptide and based on the recent Lancet paper meta-analysis, that you can already, at least at 6 months, you already see, at least on a group level, you start seeing the efficacy on C-peptide quite clearly. That might still be a bit difficult on an individual level to know if you have responded from a C-peptide perspective, because the individual trajectory of the disease is still very variable. But if you have a good, relatively good biomarker for your therapeutic, definitely, I think it would definitely make sense to use an adaptive design. But it's all about those biomarkers, and it would be nice also, I think, for going forward for the field to do similar kind of things that have been done in the oncology space, where you have like these large trials, like collaborative trials, where you have 5-10 different assets that are all in the same trial, and then depending on and you have different diagnostics, and then you can sort of you share the costs for placebo arms and like trial protocols, and then you sort of go in a way head to head, but you adapt the trial as it goes to see which asset should continue to be in the trial and which should have a lower likelihood to be in the trial.
Ulf, is this a current procedure that's in place in oncology trials?
I think one of those, I remember one trial, I think it was done in breast cancer many years ago. Well, this must be at least 10 years ago. It was done, this kind of trial. So it's usually then financed through consortia or something like this, or maybe different companies come in and because they want to evaluate their own assets in a very cost efficient manner. But that has been done and is probably being done in oncology. So where you have like use a baseline diagnostic based on like tumor like RNA expression, and based on that you have the initial randomization that the different therapeutics sort of go, but as the trial goes on you can look at the tumor response, the randomization is changed, so it's adapted based on that. The assets that seem to show a better response, they are enriched then in the randomization and that kind of stuff.
So it's not something entirely new. It's been done in other diseases, so it's just not out of the realm. And then from Diamyd, there's a platform called PanCan in oncology using the adaptive format with assets from different sponsors in pancreatic cancer. What about your direct to consumer sort of advertising for the, to get people enrolled in your trial? Have you even done anything like that, or is it really just sort of through the clinical recruitment?
Yeah, so we definitely, we do like through influencers, for example. So we have in Europe, we have worked with a number of different influencers in the type 1 diabetes space. So that's one way of obviously reaching out beyond. I mean, it's very important. The clinics, they have their own networks and their patients that they see. So that's one way. And they also have, depending on the clinic, their social media channels that they can use. But how do we, especially in this kind of case where we're going for a genetic subgroup of newly diagnosed type 1, it's even more like a orphan disease clinical trial where you really need to reach out much broader than the existing clinics. So you want to have referral from neighboring clinics if possible, all these kinds of things. So it's a lot is about social media, influencers, and reaching out through that. So there we have both influencers in Europe and also now in the US, several influencers that we are working with that also Elna knows more details about.
Yeah, we've actually had a lot of inquiries from one that was done by Man of Steel, Ben Zeal. It's great. He has a great visual in there of hitting with a baseball bat, hitting his sofa, what the attack to show the attack on the beta cells. And people have liked it. And one of our PIs just screened today, and we got another one, and all of them came from the same TikTok thing. So that it's very helpful. I work a lot with the JDRF Clinical Education Volunteers. Since we're partnered with them, there's a wonderful program that's been started to get education out about all, trials, and particularly theirs that are partially funded by them, which we're a part of. So that has been very helpful. And then we just have a lot of different materials and things for the sites to use and do things locally around the sites. But really, national awareness is what's most important here. So we're building that.
Yeah, I also have something in the chat again from Manuela. She wants to just put in a plug there for Enodia in EU might be able to help find DR3, DQ2 patients for and asking which EU countries are you running a trial in. So maybe that's like an offline conversation. But
yeah, we know them.
I also wanted. Oh, sorry.
No, we know them. Enodia, Chantal and all of them.
I think she's putting that out to others in the chat.
That's great.
And then, also I've had a couple conversations, Ulf, with scientists here in the US talking about getting the HLA test implemented in the newborn heel stick. What do you think about that? Do you think it's, first of all, possible? And then would it help, how, I guess it is possible, but how easy would it be? And then, how much would it help trials like this, like Diamyd and others that will come to identify the best populations for their drugs?
Yeah, so first of all, everything is possible, I would say. But that's definitely something that is definitely possible. I think I even did part of my PhD project working with the PKU registry in Sweden, so I have some insights into that. I think in general, to, for example, I think in the future, most likely, we will see when in the future it happens, we will all have our genome sequenced anyway. It will be cheap and simple to do, and then you have a completely different baseline to go from when you start doing, for example, clinical trials or screening programs and all these kinds of things. But I think it would make sense, if possible, to, for example, if you could sequence even the HLA part of the genome, because then you will get a baseline genetic risk for all individuals for autoimmunity itself. So that would be, and that would be incredibly valuable for example, the screening programs for type 1 diabetes, celiac disease, any other, which are already now coming. But then you would have like already that genetic risk, baseline risk, that would be valuable to say, well, should you be someone who should be part potentially of a screening program or not?
Yeah, it would be really, it would really facilitate.
Yeah. Oh yeah.
The speed of how these drugs could be developed, right?
Yeah. It would
and
be
it is interesting that the parental SCID, the community, they rallied pretty hard in the US, and due to their efforts, the SCID test was implemented into the newborn heel stick. So you have to think that maybe, if JDRF T1D breakthrough, lobbied hard, that perhaps they could get some things done there. So that would be great too.
It would, yeah. And it's the nice thing with genetics is that you only have to do that test once. It's not something.
It's not that deep.
So it shouldn't change too much over
No, it's just like it's there, and then it's there and it's useful.
Yeah.
So anyone else in the chat? And questions, comments? I think I got them all. Okay, well, great. We'll see you at the booth at ADA, and great to hear the progress from Diamyd, and thanks again for coming on and sharing. Really appreciate it.
Thank you so much, and thanks for everyone joining.
Thanks so much. Bye-bye now.
Bye.